Okay, let's talk a little bit about the cerebral spinal fluid flow and ventricles. Remember, we learned these ventricles in lab class. So we have 4 ventricles. We have 2 lateral ventricles, and then a 3rd ventricle and a 4th ventricle. So it says our ventricles are the lateral ventricles, the 3rd and the 4th ventricle. They each contain a choroid plexus. These choroid plexuses are where we have the ependymal cells that are going to make cerebral spinal fluid. It's important to understand that all 4 ventricles have choroid plexuses. I know in lab you can really only see the choroid plexuses in the 3rd ventricle, but they all have them. All right. So it says there's a blood cerebral spinal fluid barrier. So we do have a barrier so that blood and the products of blood cannot get into our cerebral spinal fluid. Cerebral spinal fluid is pretty pure. I mean, it's separated from the rest of our body. So it says substances cannot pass between cells through this blood cerebral spinal fluid barrier due to tight junctions of blood cells. All right. There's also astrocytes that form the blood-brain barrier. They form this blood cerebral spinal fluid barrier. It says cerebral spinal fluid, it's produced in our choroid plexuses of the ventricles, and it's reabsorbed between the skull and dura mater in this area called the sagittal sinus, containing these structures known as arachnoid granulations. And we'll talk about that here. So here is some flow of cerebral spinal fluid. So we've got our ventricles producing cerebral spinal fluid. It's flowing out all around the brain. It's flowing into each ventricle, flowing down around the spinal cords. But if you look, whoops, I didn't mean to do that. If you look, there is a space between our skull and brain right along that longitudinal fissure. This is called the sagittal sinus. And here's a close-up view of it. Here's the skull. Here is the brain. The meninges are right here. And we have this space called the sagittal sinus. It's in between the brain and the skull, right down that longitudinal fissure. Containing, or I should say within this sagittal sinus, are these structures known as arachnoid granulations. Arachnoid granulations contain these, these basically capillaries called arachnoid villi. And these arachnoid villi are going to reabsorb the cerebral spinal fluid at the same rate our choroid plexuses are making it. So we make cerebral spinal fluid at the same rate we absorb it. Now, if something happens to these arachnoid granulations, let's say they are not working properly and they're not reabsorbing cerebral spinal fluid the way they should, we may produce more fluid than we can reabsorb. And that's a potential life-threatening disorder called hydrocephaly. Hydrocephaly is where you have water on the brain. Hydro means water, cephaly means brain. So you have water on the brain, essentially. It's the cerebral spinal fluid. And what it does is it just fills up this subarachnoid space, fills up the sagittal sinus, and if you get too much fluid in there, it'll push on the brain. And remember, a lot of the brain's blood vessels are going to be on the surface of the brain. So once you start squeezing and putting a lot of pressure on the surface of the brain, you can shut off blood flow to the brain, and it can actually end up in death. So a person with hydrocephaly needs the fluid in this sagittal sinus and subarachnoid space to be drained. A couple of years ago, I had a student in lab, and I was going over the cerebral spinal fluid flow in lab, and I was talking about how cerebral spinal fluid flows through the arachnoid space and that kind of thing. And he raised his hand and he said that his girlfriend had an accident and her arachnoid granulations weren't functioning, and she had hydrocephaly. And what they did to remedy the situation, they can't fix those arachnoid granulations, but what they can do is they can put a shunt into her brain, or between her brain into her meninges, that drains that cerebral spinal fluid. Now, this can be temporary, so they can insert a shunt into your head to drain the cerebral spinal fluid into like a bag. That's a more temporary solution. If the brain is damaged, or maybe there's swelling and accident and those arachnoid granulations aren't doing the job, the shunt will drain into a bag. But in the case of like this fellow's girlfriend that I had in class, she had to get a permanent shunt that went from her brain. And what they do is they run it down underneath the skin into her stomach, and it would drain into her digestive system. So she had cerebral spinal fluid constantly draining into her stomach as her choroid plexuses were making it. So she could still have this balance between how much cerebral spinal fluid she makes and then how much she needs to reabsorb or drain out. So hydrocephaly is a pretty potentially life-threatening problem. So here is a good picture of these arachnoid granulations. So it says the arachnoid granulations contain arachnoid villi that are grape-like clusters that are going to reabsorb the cerebral spinal fluid. Now look at this. We produce and reabsorb about 20 milliliters of cerebral spinal fluid per hour. So you can see why hydrocephaly can be such a dangerous thing. It can kill a person within hours. So we reabsorb and produce cerebral spinal fluid at a rate of 20 milliliters per hour. Okay, so that takes care of everything I wanted to talk about with our cerebral spinal fluid. Now we're going to start wrapping up the chapter. I want to talk about hemispheric lateralization, talk about how the different hemispheres of our brain control different things. And then we're going to get into some disorders, and then we'll be finished with the chapter.